DNA requesting duplicate. Do you copy? Well, maybe the more pressing question is, how do you copy? Well, you got these things called purines, adenine and guanosine, and you've got pyrimidines, thymine, cytosine, and uracil. Purines and pyrimidines pair together. A matches to T, and G matches to C. Pointy letters, TAAT, round boys, GCCG. Kind of a Kiki Booba situation. You stick them together, and that's a Watson-Crick pairing, baby. And this is gonna become our DNA. But what do these pairings mean? What is it? Well, if you chop up DNA into its little pieces, it looks like this. Oh my god, chemistry! Scary! Whoa. Whoa there, it's cool. It's alright. Here's a fun little titty bit. DNA stands for deoxyribonucleic acid. I know it's a big word, but it's descriptive. RNA in the same vein is ribonucleic acid. DNA is literally a ribose sugar deoxide, unoxied, no oxygen, right there. Nucleic acid. This is a nucleotide. Here's another neat thing. ATP, you know ATP. Okay, the one thing you learned in high school bio, say it with me. Mitochondria is the powerhouse of the cell. Yeah, ATP is the energy currency. Bio cash money. Money is power. Chemical energy power in this context. ATP stands for adenosine triphosphate. There was another A that I mentioned earlier. Adenine. Yeah, it's the same A. The same. Except it's technically the RNA one, but for DNA we just scribble that out and deoxy it, and yeah. Adenine, adenosine, tri-3-phosphates. You must be wondering if there's an ATP. Is there a GTP, a CTP, TTP, UTP? And the answer is yes! Yes, yes, and yes. To make DNA, we link them all together with phosphodiester bonds. This is a phosphate, and this is an ester. Diester means that there's two of them, so the bond is two esters with a phosphate in the middle of it. Another rule in DNA is that it's always built from 5' to 3'. We number the ribose sugar like this, and the reaction happens in the 3' position to add the next nucleotide. We gotta be kind of funny about it if we're doing it any other way. Bringing us to how it's actually replicated. First we've got to unzip it, and it's unzipped with helicase, but unzipping causes strain, so to relax it, we've got topoisomerase. DNA is a double helix, so helix A's, suffix A's is an enzyme, proteins that do stuff. Helicase opens up the helix, and topoisomerase, topography is the way that physical features are arranged, and isomers, different chemical arrangements of the same atomic components, and the suffix A's, again, denoting enzyme. So DNA remains the same, but it's in a way that's more relaxed as the helicase opens it up. The helicase opens up the DNA, but for what though? Well, to put this thing on, the ribosome, or as I like to call it in my head, the DNA replicatinator! Okay, but the ribosome is probably more proper when stuff with the suffix "-some" describes something made of more than one protein component, like ribosome, 2 pieces, proteasome, 3 pieces, apoptosome, see? But the ribosome, this big guy, is made up of, well, a couple of parts. There's the clamp and the clamp loader. The clamp loader loads the clamps. And think of the clamp as a handcuff for keeping the more DNA-making mittens on the template DNA. Said mittens are polymerases. They're the part that add on the nucleotides. Polymers are a chain of many molecules linked together. There's 2 relevant kinds of polymerases. Pol 3 gets the prime real estate of the leading strand, and it can go on continuously. No stopping it, just let it go. While Pol 1 gets the lagging strand. Why does it lag? Well, remember when I told you guys that we always build from 5' to 3'? Yeah, well, DNA matches up like this. 5' to 3' matching to 3' to 5'. So the 5' to 3' strand is technically asking the Pol 1 to write backwards, to write 3' to 5', which it can't really do. Think of how we typically write from left to right. It's weird to write right to left for anyone who's not Hebrew, Arabic, or East Asian writing vertically. Polymerases always add nucleotides 5' to 3'. If you're asked to write the stuff, sit on the 3' end and pretend you're the polymerase and write 5' to 3'. When the template is 5' to 3', like on the lagging strand, the polymerase needs to work in pieces. These pieces are called Okazaki fragments. These are set up by primers, RNA primers. Remember, as this is only in the case in DNA that we'll use primers to get started. DNA needs to be primed to be ready to replicate. When DNA is built, the primers are replaced with pol-1 exonuclease activity. Exonuclease eats from the ends of DNA as opposed to endonuclease, which can eat the DNA from the middle of it. After the primers are replaced with proper DNA, then ligase comes in. Ligate means to stick or put together, not to be confused with lysate, which means to break or take apart. Ligase seals the spaces between the Okazaki fragments. And then you've got replicated DNA! Hooray! For prokaryotes, single-celled stuff like bacteria. But the only difference in machinery between us and germs is that instead of pol 3, we have pol epsilon. When they have pol 1, we have pol alpha and delta. Pol alpha acts like primase, and then delta takes over the work. We also call helicase MCM, the clamp loader RFC, and the clamps PCNA. Pretty much everything works the same way as the prokaryote, except everything has worse names. And that's how we have new DNA. Replicated. Hooray! Bio always has a lot of stuff going on, and it's really rough if it's just memorization, but if you keep in mind that the stuff here kinda names itself, and try to understand why it works the way it does, it gets easier. Life always copies and tweaks things ever so slightly with every iteration, and how it all comes together is a story that builds on itself. Looking at prokaryotes and seeing how it still has a lot in common with eukaryotic cells that set us up, in addition to other animals, is kind of the equivalent of looking at a rock to figure out what's the most important features to make a mountain. And also that rock DNA is highly conserved, because the consequences of not copying it right can be problematic for subsequent cells made with that DNA. Luckily, the replosome fairly rarely messes up, about 1 in a billionth when it comes down to it. And even when it does make a mistake, it usually gets fixed before it becomes an issue. How does it get fixed? Well, that's a story for another time. Anyways, thank you for listening!